Today I'm going to tell you the story of how a beetle came to use a small molecule made by a bacterium to fight fungal infection, and how we studied this system even though we couldn't culture the bacterium in the lab. So this is Lagria villosa, a soybean pest mainly found in Brazil. It lays its eggs in soil in these clutches. Now you may be aware that soil contains all sorts of microbes, including many fungi that can act as pathogens to the beetle eggs, preventing them from developing. However, our collaborators Martin Kaltenpoth and Christian Hertweg found that the eggs contain a compound called lagriamide that reduces the frequency of fungal infection in eggs. Now, an extremely interesting aspect of this lagriamide structure is that it is actually very similar to another known natural product, bistramide A. These parts are the only differences between bistramide and lagriamide. But bistramide doesn't come from a beetle at all, but a marine tunicate. Another thing to note here is that both structures look more like bacterial compounds than anything known to be made by animals. So who makes lagriamide and bistramide? It seemed likely that in both cases these compounds are made by bacterial symbionts. So Martin's lab extracted DNA from beetle eggs and sequenced just the 16S ribosomal RNA genes, which tells us what bacteria are present. And that told us that the eggs are always associated with a number of strains of Burkholderia gladioli. So these were prime candidates for the producer of lagriamide. So Martin's group attempted to isolate B. gladioli from the eggs and were able to get one strain they called LVSTA. And our other collaborator Christian Hertweg was able to isolate a number of compounds from this strain. One of these compounds is even antifungal, but you will notice that it doesn't produce lagriamide. And through sequencing the genome we can tell that LVSTA does not possess a pathway capable of making lagriamide. 16S sequencing also showed that LVSTA is not always present and doesn't correlate with lagriamide presence. A number of other B. gladioli strains are more consistently present, but unfortunately Martin's group were never able to isolate the lagriamide producer. So it began to look like the lagriamide producer might be unculturable, like many symbionts. But why is it that symbionts are often difficult to culture? Well, symbionts start off as free-living bacteria, and at some point become stably associated with another organism, such that they never spend any time apart. If the host organism provides a lot of metabolites, then there is simply no evolutionary pressure for the bacterium to keep genes around that make those same metabolites. So while free-living bacteria have fairly large genomes with all the genes they require for independent life, symbionts have much smaller genomes with many genes missing. As more and more genes are lost, the genomes of symbiotic bacteria eventually don't have enough genes to allow them to be grown independently. But how do we study such bacteria that cannot be grown? Well, one way we can find out about such organisms is by doing culture-independent sequencing, also called metagenomics. For instance, if we have a whole bunch of bacteria that live inside a beetle, but none of them can be grown independently in the lab, we could just extract DNA from the whole insect. But this, of course, would result in a complex mixture of DNA from the insect or any one of the bacteria present. And so when you sequence this mixture, there are a number of challenges in data analysis. 1. We don't know how many species there are. 2. The species that are there could be completely unknown to science, and so we can't just align sequences to genomes in a database. And 3. We are sort of randomly sampling when we are sequencing. So if a species is really rare, then we have less chance of getting all the parts of its genome, unless we increase the total amount of sequencing we do. In other words, metagenomes require much more sequencing than isolate genomes. So one of the areas of research in my lab is figuring out ways of analysing metagenomic sequence data so that we can get past these challenges and separate each of the genomes in the sample. And we've actually devised our own computer program called Automator, which we hope will make this task easy. We've made this available on GitHub, and you can also read our paper on it. And I'll just go through how we used it to process the Lagria villosa metagenome. So we took some Lagria villosa eggs and did completely random sequencing. After the sequencing reads were assembled, we got 28,000 contiguous sequences that we call contigs. And here I'm representing them as dots, with the diameter of the dot scaling for contig length. Now we can begin to make sense of this by looking at a number of parameters. Here I'm plotting them on scales of GC% versus coverage. We know that different species have genomes with different GC% versus AT, And also in a complex mixture, we would expect different species to have different abundances, which is what coverage is. It is basically measuring the abundance of each contig. But what we can also do is to find where the protein coding genes are in the contigs and look at how similar they are to known genes in sequence databases. And that can tell us whether they are more likely bacterial or eukaryotic or whatever else. And this is important because we have a lot of eukaryotic sequence here, likely from the insect genome. Here we are colouring the contigs by kingdom. So Automator can get rid of all contigs that are not bacterial, and this makes the data much simpler. If we change the colouring from kingdom to family, you can see that it is actually quite difficult to separate out individual genomes on these axes. But one thing we do know is that nucleotide composition is different in different strains of bacteria. What do we mean by that? Well, if we look at the sequence of any contig, what we can do is split this into what is known as kmers, where k is some arbitrary small number. We'll call this first kmer K1. Kmers overlap, so K2 will be one nucleotide along. With k equals 4, we have 256 possible kmers, although we don't count reverse complements separately. But to give ourselves more resolving power, we use 5mers, of which there are 1024 possible sequences. So when we want to compare the 5mer composition of one contig versus another, we have to total up the times each occurs in that contig. But it's very difficult to really understand how different contigs are in 1024 dimensions. The solution is to put this data through a process called dimension reduction, which aims to represent the variance in all 1024 dimensions in just 2. To illustrate what I mean, let's go back to our GC coverage graph, with only bacterial contigs coloured again by family. We could say here that we are looking at just one dimension of nucleotide composition, the percentage of GC, and it's hard to separate things based on that. But if we put the 5mer frequency through a dimension reduction algorithm called Barnes-Hutt stochastic neighbour embedding, you can see now that there are many discrete groupings. You can think of the distance between points in this two-dimensional graph as being roughly proportional to their closeness in nucleotide composition, and contigs from the same genome should cluster together. The next step is to use a clustering algorithm called dbScan to group the individual points into discrete groups. This algorithm looks at the local density of points, and the EPS parameter tells it how far away to look for density. As we vary EPS, we will assess the clusters by the presence of certain genes, known to occur only as single copies in bacteria. The number of these genes we see in each group, out of the ones we expect, tells us how complete the group is, and the number of genes that are found only once in a group tells us how pure it is. We only consider good groups, which are over 20% complete and 90% pure. And as a measure of how well we are doing, we look at the median completeness of those good groups. We start with a very small EPS, which will give us very small groups and gradually increase. Here we are only colouring the groups that are over 20% complete and 90% pure. At the end, we find that an EPS of 2.0 gives us the highest median completeness. You will notice that these results are not the highest number of groups possible, but it gives us the most complete ones, and there are a lot of contigs left. But that's okay because we will take out the coloured groups and redo this procedure. What we find is that dbScan generally finds the best separated groups first, and then in subsequent rounds it separates more difficult cases. And we carry this on until we can get no more good groups out of the data. At the end of that process, here are the groups that dbScan identified in the Lagria villosa egg metagenome. But actually we don't just use the two BHTSNE axes for dbScan. In order to separate further, we use coverage as a third dimension. And in this particular case, we could see that there was one genome with almost 2000x coverage. This genome appears to be an uncultured bacterium we call Burkholderia LV-STB. Almost the same coverage is this one contig that contains a complete trans-AT polyketide synthase pathway that we think makes ligriamide. But this contig is in a completely different part of BHTSNE space. Through single-cell genomics, we found that this contig is actually part of the LV-STB genome, and it's likely on its own because it was recently horizontally transferred to this genome. It's just that the nucleotide composition of the pathway hasn't normalized to the rest of the genome yet. We call the ligriamide pathway LGA, and it is almost 90 kilobase pairs long, consisting of a number of genes shown here that are clustered together. It is what is known as a trans-AT polyketide synthase pathway. Now, I don't have time in this talk to go into much detail about how this type of pathway works, but I'll just say that they typically have very large genes, like this, that consist of many different enzymatic domains that are covalently connected through the primary amino acid sequence. We split these into modules, which each add a unit of malonate to the molecule. Malonate is added to the polyketide product as acetate, and the rest of the molecule is released as carbon dioxide. Acetyltransferases, or ATs, load an activated form of malonate, a thioester with coenzyme A, onto acyl carrier proteins, or ACPs. Ketosynthases, or KSs, accept the substrate from the previous module and add it to the next malonate through a Claisen condensation. In the process, carbon dioxide is released from the malonate. The resulting tetrahedral intermediate is released from the KS when the O- becomes a carbonyl again. If we just have a KS and an ACP, the structure is left as a beta-keto. But that isn't the end of the story, because you can have other domains to further tailor the structure. If we additionally have a keto reductase, or KR, then we get the beta-hydroxy compound. If we have a dehydratase, or DH, then we have an alpha-beta double bond. We could also have a enoyl reductase, or ER, to get the fully reduced beta-carbon. And we can also have a C-methyltransferase to give the alpha-methyl compound. This is not all the possible combinations that you can get in a module, but it hopefully gives you some idea of what can happen. So with that, let's look again at the LGA pathway. Now, using what we know about this type of pathway, we can predict the structure of the intermediates covalently attached to each module. And we see that these predicted structures fit really well with the known Lagria structure. After the molecule is cleaved off from the PKS, there are just a few more structural conversions that would need to happen, and these can easily be explained by the presence of additional genes in the pathway. Although we don't really know how the spiropyran structure is made. But overall examination of the sequence of the pathway fits so well with the known structures that this pathway is extremely likely to make Liguria. So let's do a quick recap of what we know so far. We know that the LVSTB strain is consistently associated with the beetle Liguria villosa. But we also know that other related strains are often present. We also know that Liguria, which protects the beetle eggs from fungal infection, is made by LVSTB. This all leaves us with a question: how and why did this complex situation evolve, where we have multiple symbionts even though one seems to be the most important? So one of the first questions we asked was whether LVSTB was unique in the microbiome in its ability to biosynthesize a complex small molecule. Here are the different genomes we could assemble from the Liguria villosa egg microbiome, along with their taxonomic family. Here I'm showing the size and type of biosynthetic pathways for small molecules in each genome, and you can see that many have larger and more pathways than LVSTB. Here I'm highlighting the LGA pathway for Liguria. LVSTB actually has some other pathways as well. Remember though that previously Martin's group isolated another strain of Burkholderia they call LVSTA. Although we didn't find it in this microbiome, we'll include it here for comparison. We already know that this strain produces a lot of compounds, so it is not surprising that it has so many pathways. And here are just the pathways from the metagenome and LVSTA that are larger than 50 kilobase pairs. You can see that there is the potential to make compounds perhaps as complex as Liguria. So what this may mean is that there is a sort of reservoir of bacteria that can make small molecules associated with this beetle. Most of them are minor members of the microbiome, but if environmental pressures change and one of those compounds starts to give a survival advantage, then selection could favour that species. In other words, perhaps having these different options is useful for the beetle so that it can adapt to different environments. You may wonder then, what is unique about LVSTB in this microbiome? Well, one of the questions we asked was which genomes in the microbiome are reduced. Because genome reduction takes time, that would suggest that the reduced symbionts might have been with the beetle for a long time. We now look at the total length of our genome sequences and compare them to LVSTA at the top because it was obtained from a cultured isolate and we know it is complete. You can see that many of the microbiome members are small, but many free-living bacteria have genomes much smaller than LVSTA. So perhaps what we should be asking is how large are these in relation to their closest relative? So here we are plotting the size as a percentage of the size of the closest relative. And we can also alter the graph to be in descending order of percent size. And you can see that some bins are quite small, including LVSTB. But these bins could be small either because they really are reduced or they are larger and just have not been completely assembled. How do we tell these two possibilities apart? Well, when we run Automator, it estimates the completeness of a genome based on the presence of 139 single copy marker genes. And that is what I'm showing here. However, this doesn't tell us whether the genome really is incomplete or not, because there are some genuinely reduced genomes that we know are complete that show low completeness by this measure because they have lost some of those genes. However, there are some genes which are present in even very reduced genomes. For example, genes that are required for protein synthesis that must be present. We call them core genes here. These genomes at the top of the graph are large genomes that are also complete by both single copy markers and core genes. Other genomes are small and not complete by both measures, and so we think they are not reduced, but rather are not completely assembled. But only the LVSTB genome is small, but near complete in terms of core genes. We interpret this to mean that it really is reduced, otherwise it would be missing more of the core genes. So we know that LVSTB has a reduced genome, but what genes is it missing? Well, we looked into this in a lot of detail, and if you want to drill down into all these details, you can have a look at our paper. In terms of metabolism, we are missing a lot of the genes involved in making thiamine, riboflavin, nicotinate, pantothenate, vitamin B12, biotin, aromatic amino acids, serine, methionine, glycine, histidine, cysteine, glutamine, arginine, chemotaxis and flagella, glycolysis, and nucleotide biosynthesis. Genes are also missing in basically all the DNA repair pathways. This is actually quite common in reduced genome symbionts, and it leads to an increase in the speed of genetic drift because mutations are not repaired as efficiently. Because mutations accumulate more quickly, non-functional pseudogenes are also generated quicker for genes that do not have a tremendous amount of selection pressure on them. So the next step was to see which genes in the LVSTB genome are conserved in its closest relatives, which are strains of Burkholderia gladioli. One key part of this figure is that few of the genes that are found universally in B. gladioli are retained in LVSTB, but there are also some genes which seem to have been acquired only by LVSTB and not its relatives. So the question that arises here is, when did these arrive? Well, it turns out that it is possible in some circumstances to estimate the age of horizontally transferred genes by looking at how the nucleotide composition is different from the parent genome. This was figured out by Lawrence and Ockman in 1997. They reasoned that when a gene has just been transferred from one species to another, its nucleotide composition would look like the donor genome, and over time this would slowly normalize to look more like the recipient genome. This process is called amelioration, and it takes hundreds of millions of years. But they reasoned that the first, second, and third codon positions would change at different rates because of the differences in degeneracy of the amino acid code at those positions. They worked out this equation, where delta GCHT is the change of GC content in horizontally transferred genes, while GCEQ is the GC content of genes at equilibrium in this genome, and GCHT is the current GC content of genes that have been horizontally transferred. S is the substitution rate, basically the rate of mutation, and kappa is the ratio between transition mutations, where a purine or pyrimidine is converted to a nucleotide of the same type, and transversion mutations, where a purine is changed to a pyrimidine or vice versa. Now, it is known that there is a relationship between the GC content of a genome and the GC content at the first, second, and third codon positions in the component genes. These relationships are shown as straight lines on this graph. What this means is that first, second, and third codon positions of the native genes in the genome appear on these lines, but the different codon positions of horizontally transferred genes do not. But we can use our equation to work out how the GC content of each codon position in these genes changes as we go back in time, to a point at which they meet the lines again. These points now represent the GC content at each codon position at the point in time when the gene was received through horizontal transfer. By counting how many iterations of the equation we go through until we get the least error between the points and the lines, we can estimate the time since the genes were transferred. Using this method, we could construct a timeline of horizontal transfers to the LVSTB genome. Now, we don't know the exact mutation rate in LVSTB, so we use some estimates from other symbionts. So the exact numbers here are probably not that accurate, but the important thing is that the relative order of acquisition should be right. The oldest acquisition is this group of phage genes, and we're not really sure why those have survived so long. But the next oldest is the Liguria pathway, and this is the oldest acquisition that is potentially useful to the symbiosis. We propose that this was acquired by a free-living ancestor of LVSTB before the symbiosis was established. That would mean that genome reduction happened after that. With that being said, the weird thing here is that when genome reduction was happening, it appears that LVSTB was able to pick up a lot of other genes, and a lot of them appear to be adaptive for ongoing genome reduction. For example, we have a lot of transporters for things that LVSTB can no longer make. There are also some DNA repair genes, and perhaps these additions compensated somewhat for the ongoing losses of these genes in the genome. The most strange thing about all this is that in a lot of models of how genome reduction evolves, it is implicit that there will be a degree of genetic isolation which aids this process. Here we have a symbiont that has access to a complex microbiome and is apparently able to take up genes. However, the incoming flow of genetic information has not been enough to prevent genome reduction. Supporting the idea that the symbiosis has existed for a long time, the beetle has evolved special structures to hold bacteria. In Lagria hirta, the ovipositor is surrounded by special structures, shown in red here. The Burkholderia symbionts were detected in these structures by fluorescence in situ hybridization, or FISH. The placement of these structures means that the eggs can be coated in the symbionts as they are laid. Washings from the eggs show the symbionts by FISH. When the larva hatches, it has special structures, shown in red here, which also hold the symbionts. The symbionts can also be visualized here by FISH. And we can estimate when these structures evolved based on a combination of molecular methods and the fossil record. This is a previously published time-calibrated tree, and out of all these beetle species, only Lagria hirta and Lagria bryanti have these special symbiont chambers. The common ancestor of these two species existed 55 million years ago. The closest relative that doesn't have the structures is Adenata brevicolis, and the common ancestor of all these existed 82 million years ago. So from this we can date the evolution of the structures between 55 and 82 million years ago. We can also look at the phylogeny of Burkholderia gladioli and strain LV-STB. You can see that the closest relatives to LV-STB are all B. gladioli strains, but that LV-STB is very divergent from them. We can look at this divergence, and using the same estimations of mutation rates we used earlier, we can calculate that the common ancestor of LV-STB and B. gladioli existed between 6.2 and 49.8 million years ago. Now, because the mutation rates are really uncertain, the time estimate is a really large range. But because the common ancestor can't have had a reduced genome, that means that genome reduction happened after the symbiont-bearing structures evolved in the beetle hosts. So here is a sort of very approximate timeline of what might have happened. The common ancestor of all these beetles with Burkholderia symbionts likely came into contact with strains of Burkholderia through their plant diet, because many strains of Burkholderia gladioli are plant pathogens. Perhaps the beetles that ended up with the bacteria coating their eggs had a greater survival rate, and so over time they evolved special structures to ensure that this happens. After that point, the genera Liguria and Liguria diverged from each other. According to our estimations, which I should remind you are very approximate, the divergence of LV-STB from the rest of B. gladioli happened after that. Relatively more recently, the horizontal transfer of the Liguria pathway happened. Because the symbiosis seems to be predicated on Liguria and the pathway genes are the oldest and only genes that seem to provide a benefit to the host, we think that genome reduction happened after LGA was acquired. In that time, LVSTB became dominant in the microbiome of the host, like it is today, and was more consistently vertically transferred, while other strains maintained their sporadic distribution. And when we look at the present-day genome of LVSTB, it seems likely that it is largely dependent on the host. If it needs the beetle to thrive, that might explain why it is more efficiently vertically transmitted compared to the other symbionts, which do not need to live in the beetle. So that is the end of the story, although we're currently trying to confirm some of these evolutionary details by looking at other host beetles and doing comparative genomics. Before I finish, I should acknowledge first my lab members. In particular, Sam Waterworth is the postdoc that did all the work on the Burkholderia symbionts I presented today, while my former student Ian Miller and current students Evan Rees, Sid Uppel and Kyle Wolf worked on Automator. We also worked with Laura Flores, Christian Hertweg and Martin Kaltenpoth on the beetle system, and Frederica Ray helped provide validation data for Automator. Sam is funded by the Moore Foundation, and work on Automator is funded by the NSF.